You don't all need.
Sure, sir. Bismillah ar-Rahman ar-Rahim. Assalamu alaikum and good day, ladies and gentlemen. I'm Hazima Mozam and I welcome you to online certificate course on basic plant tissue culture techniques organized by Comstech in collaboration with International Center for Chemical and Biological Sciences and Pakistan Biotechnology Information Center, Karachi, chapter Karachi. This course is designed to address the interest of the scientists and research scholars working in the field of plant biotechnology, including genetics and molecular biology. This course will be delivered by some of the very renowned experts in the field internationally. With this, we will begin today with the recitation from Holy Quran.
Bismillahir Rahmanir Rahim.
In the name of Allah, the entirely merciful, the especially merciful.
Ar-Rahman.
The most merciful.
Allama al-Quran.
Taught the Quran.
Khalaqa al-Insan.
Created man.
Allamahu al-Bayan.
And taught him eloquence.
الشمس والقمر بحسبان.
The sun and the moon move by precise calculation.
والنجم والشجر يسجدان.
And the stars and trees prostrate.
والسماء رفعها ووضع الميزان.
And the heaven he raised and imposed the balance.
صدق الله العظيم.
Jazakallah. With this, I would request His Excellency, Professor Dr. Iqbal Chaudhry, Coordinator General Comstech and Director ICCBS for his welcome address. Over to you, sir.
Okay, so I think we need to rely on audio only because there is something really wrong in turning on the camera. Please go ahead, please.
Sir, thank you very much. As explained earlier, this course will be delivered by some international experts in the field, from the field of plant biotechnology. We have Professor Dr. Panjaman Azad with us. We also have Dr. Muhammad Usman and Dr. Muhammad Iftihar Ahmad. With this, I would request you, His Excellency, Professor Dr. Iqbal Chaudhry, Coordinator General Comstech and Director ICCBS for his welcome address. Over to you, sir.
Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullahi wa barakatuhu. I would like to welcome you all in this very important webinar, which is focused on a technique universally used, globally used for a variety of different purposes. The tissue culture technique has a wide range of applications in biomedical sciences, in toxicology, agriculture, horticulture, and many other fields. So developing a skill in tissue culture, as well as understanding the basics, nuts and bolts of tissue culture, is extremely important for a good research project, as well as for initiating number of important projects. So this is what this course is all about. The certificate course is primarily focused on plant tissue culture. We all understand that plants are the fundamental component of human life. Agriculture is based on plants of different kinds, and propagation of plant has been always a very major challenge, especially the one which requires long period. And in that case, new technologies have been developed. So plant tissue culture has been a very good technology, a very practical technology, which is now used far and wide. It was initiated at the laboratory level, but now you would find that farmers in different countries of the world are capable of doing plant tissue culture by themselves. So this technique has gone beyond laboratory and research paradigm into practical basic use. What primarily you're using this nature's capacity that plant can be regenerated from any part, from roots, from leaves, from stems, and different parts of the plants. And you can literally produce thousands and thousands of copies of identical plants by using tissue culture technique. Now think of it that if you can use this technique for the production of plants which otherwise take several years, you add so much of economic value to the plant sciences as well as in agriculture. And then on top of it, seeds are often infected with different kinds of diseases, and plant tissue culture provide you disease-free, virus-free germplasm for propagations. The technique has lots of significance. We have been using plant tissue culture in our institute in Karachi since long, and we have developed a number of products, horticulture and floriculture, and this is very doable. And I'm glad that this is one important area in which comstech is trying to develop practical training. So many of the initiatives of comstech are focused on development of hands-on skills, which are required apart from high technology lectures or discussing about the basic fundamental sciences or research topics. It is extremely important that we focus on the development of basic skill set, which people can use in their own practical work and can apply for economic development. And this is what this certificate course is all about. So I'm extremely grateful to my colleague, Professor Dr. Samar Yusuf, my colleague, Hazima Muazzam, and the resource persons of today's certificate course. They all are very competent practitioner of tissue culture. They know the science from the basic core of it as well as the practical aspect of it. So a lot of the strength are there in this course, and I'm sure that over 200 registered participants from different countries would be able to benefit from this important certificate course. With this, I would like to welcome you all, and we expect from comstech that you would be able to benefit out of this very good exercise. Thank you very much.
Thank you very much, sir, for your kind words and appreciation. With this, I would request our audience to please download a manual which I have shared in the chat box. This manual has been prepared by our organizers for this training course. With this, we will start our technical session. The first talk of our technical session will be delivered by Professor Dr. Pajman Azadi. Dr. Azadi is a Deputy Director General for Technology, Agriculture Biotechnology Research Institute of Iran, and he will be delivering his lecture on commercial plant tissue culture for horticulture plants. With this, I would request his Honorable Dr. Panjman Azadi for his lecture. Over to you, sir.
Good morning, everyone. Can you hear me?
Yes.
Yeah. I'm very pleased to have this opportunity to speak to you for this important webinar on plant tissue culture techniques. And also, I would like to thank organizer for inviting me. First, I would like to briefly introduce my background. I'm a researcher at Department of Plant Tissue Culture and Gene Transformation, ABRI, and Deputy for Technology Transfer. Also, I'm a technical manager of several commercial plant tissue culture companies for rootstock, horticultural plant rootstocks, ornamental plants, medicinal plants, and also the founder of first plant tissue culture accelerator in Iran. Okay, Agriculture Biotechnology Research Institute is founded in 1999, and after that we have established several branches in different fields of the food biotechnology, animal biotechnology, and also medicinal plant biotechnology in different parts of Iran. And Karaj is the main office in Karaj, very close to Tehran, capital. We have a different department in the office in Karaj, which is like system biology, molecular physiology, genetic engineering, microbial biotechnology, plant tissue culture, and nanotechnology. You can see here the human resources in ABRI, about 50 scientific staff. And today I'm going to talk in 4 parts. The first part is the basic understanding about the commercial plant tissue culture. Second part is application of plant tissue culture in different area. And the third part is role of plant tissue culture accelerator. And finally, some ABRI achievements in plant tissue culture. Okay, we are all familiar with the global challenges, the climate change, the problem of crop loss by pests, the water shortage, growing population, less arable land. These challenges are very important and critical for the human in the future. And of course, one of the solutions for this, to solving some part of these challenges could be agricultural plant biotechnology. As you can see here, different categories of the global biotechnology industry. Biopharma is the most important one, and then industrial biotechnology. But as you can see here, agriculture biotechnology also is very important area of the biotechnology in the world. The market size for biotechnology is about $300 billion, and more than 11,000 businesses are active in this field, and with 880,000 employments. And as you can see here, the bioagriculture during the last 2 decades increasing, the growth rate is increasing, and we are sure that in future also the importance of the bioagriculture will increase. Among the important application of the agricultural biotechnology, plant tissue culture is one of the basic and important application. The first report for plant tissue cultures goes back to the 1902 when Haberland introduced the hypothesis of totipotency of the plant cell. And after that, the technique of plant tissue culture is well translated from the concept to the commercialization. You can see here different discoveries in plant tissue culture since that, like haploid culture, in vitro mutation breeding, synthetic seed productions, and also finally gene transformation. One of the interesting example of the application of plant tissue culture, you can see in this article that the researcher could succeed to regenerate the plant from 30 years old fruit tissue buried in Siberian area. And from this fruit tissue, they succeeded to get the whole plants. Plant tissue culture has a different application, clonal propagation, or covering for self-sterility, somatic embryogenesis, genetic transformation, haploid, polyploid production, and also secondary metabolite production. And these are the main application of plant tissue culture. However, as I will discuss later, mainly in developing countries, they focus on only on large scale multiplication. In case of the mass propagation or micropropagation, there are different stages. And the first stage is the sterilization of plant tissue, and then initiation of the culture in sterilized media under the laminar air flow, and then multiplication, which usually like repeat 10 to 15 times. Each time is like about 1 month, takes 1 month. And we have a proliferation stage in this step, and then finally rooting, and then transferring to the soil, which is hardening or acclimatization. But we also have a meristem culture, which is very important for the horticulture industry for production of virus-free root esters, and we use them to produce the like certified root ester using the thermotherapy, and then meristem culture usually less than 1 millimeter meristem, and then checking by like molecular techniques to be sure that this plants is really virus-free plants. And then from this plant, we are going to have like a basic material for the horticulture plant. Also, another application is embryo culture, which usually we use it when we have some difficulties for crossing and producing F1 plants. So to overcome immaturity in seeds, usually we use the embryo culture. And one of the important application of plant tissue culture is hunter or microspore culture, which usually we use to produce inbred line as a parent of the F1 hybrid seeds. And protoplast culture is another application which is important for producing the new variety with protoplast fusion. And recently, by developing genome editing, is very important as a basic tool for the genome editing. And in case of genetic engineering, gene transformation using agrobacterium, of course, we need the plant regeneration system to be well developed. Otherwise, these techniques, agrobacterium transformation or bialistic, cannot work. So for each tissue culture technique, we need to have a optimum medium for that. And each medium is contains salt, inorganic salt, carbohydrates, vitamins, plant hormones, and finally solidifying agents. For microelements, as you can see, there are several important microelements should be included in the medium. Microelements as well. Sugar usually we use in commercial tissue culture, sucrose, or the sugar we use in the normal daily life, which is cheaper than the commercial one, or the produced by companies. Vitamins, different vitamins like vitamin B1, B6, myo-inositol, activated charcoal, plant growth regulators, and also or gerite. As you can see here, different media is used in different plant tissue culture. One of the most popular one is MS microscope, which different plants, different genera can regenerate in this medium. Of course, we need to sometimes change the media to another medium, and sometimes we need to modify each composition of each medium. In case of plant regulators, we know that cytokinins, auxins, gibberellins, and abscisic is very common to use in any plant tissue culture laboratories. In case of the cytokinin, the most common one is BA, 2IP, kinetin, TDZ, zeratine, which all they work for adventitious shoot formation, promote cell division. But for commercial use, usually we use the BA, sometimes kinetin, 2IP. We don't use that much zeratine because it's very expensive. And in case of the TDZ or TDS, we don't use it that much. Very rare to use it because of this hormone is very aggressive and could produce variation or genetic mutation in the plants. And usually, as Professor Chaudhary mentioned, we need to have the identical plants, true to type plants, and not mutants. So then we don't use such hormones in the commercial one. And in case of auxin, as you can see, IDA, 2,4-D, NAA, PPR, dicamba are the main auxins we are using in plant tissue culture. Usually they promote adventitious root formation and also induction of the somatic convergences, cell division. However, in commercial use, again, we mainly use IBA because it's more stable than IAA, and sometimes NAA, but not 2,4-D because 2,4-D is also another aggressive hormone, which may cause genetic variation. So I just want to mention that the method of the working in commercial plant tissue culture is totally different with the research use, because in research the main aim is the production of the shoots, a high number of the plants, but in case of the commercial production, of course we need to have a high number of plants, but they should be true to type, not mutant. And usually interaction of the auxin and cytokinin can result for the purpose we need. Like, for example, if we want to have a root formation in protein, we need to have a higher concentration of auxin, or for monocot, for callus induction, we need to have a high concentration of auxin. But in case of the callus induction in dicot, we have to use low concentration of auxin and plus with cytokinin. So depending on the purpose we need, we can use different concentration of different hormones for plant regulators. For example, in Anthurium, when we want to get the shoots, organogenesis shoot, we can use low concentration of the auxin plus high concentration of the BA. But when we want to get the somatic convergences plants, we need to use high concentration of the 2,4-D and low concentration of the cytokinin. So depending on the aim and the purpose we have, we can control the propagation method. GT is one of the most important for commercial production. Actually, for shoot elongation, we use it, but it should not be used in high concentration, because if you use it several times, it is going to have the abnormality in plants. And for agar as a solidifying agent is very important also, because it not strongly react with the media. But if you use the gel and gum or gel-right or feta gel, you should be careful that, because this solidifying agent may cause the hyperhydration. As you can see in the right side, the hyperhydration can cause the abnormality in the plants, and usually you cannot get good percentage of the acclimatization at the final stage. So several times using of the gel-right in the commercial use is not good actually. Activated charcoal is another important compound. Usually we use it to inhibit the phenolic or toxic compound and also for rooting. But we should be aware when we use the activated charcoal, we need to increase the concentration of the hormones at least 2-3 times because it's absorbed by activated charcoal. And when we are doing the tissue culture, one of the most important items is in which stage to take the explant. For example, in rose, as you can see, the dark circle showing the high regenerative explants. But when the plants bloom, fully develop the flower, the regeneration will decrease significantly. So for each plant, we need to know which step of the plant growth is more suitable for taking the explant. And also which part of the plants is very important. For example, in this photo, you can see when we go to the cotyledon, near the cotyledon, we are going to have a higher shoot induction. And when we go to the leaf, the number of shoot induction significantly decreased. So we need to know which part of the plant is suitable for our target. In case of the commercial plant tissue culture, we need to, for example, of course, we need some laboratory equipments like laminar flow, autoclave, incubator, pH meter, and production management is very, very important. And that's why it's very different working on commercial scale compared to the research scale. When you work in the commercial scale, you need to produce the prototype plant. And for this target, you need to have to know the original, to use the original material. And also the multiplication rate should be controlled. You cannot do subculture more than 15 times or more than 14 times. Or you cannot use high concentration of the aggressive hormones. Even like normal, like BA or IBA, PGR should be used usually less than 1 milligram per liter. So this is important because when later I will explain that we had some issues in Iran, in this case, that some like a problem, lawsuit cases for some companies. And another important thing is the time of the delivery is very important. You should be completely know about the safety margin of the growth factor in your laboratory. Otherwise, you might lose a lot of plants, tissue culture plants. And the most important item is you should produce in like a competitive price, but without losing the quality. It's very important. Therefore, you should control the disease like fungi, like bacteria in the commercial scale. Is very, very important item. So the price for commercial production of the in plant tissue culture mainly is for labors. So about 60 to 80% of the cost is belong to the labor, and the rest are for the electricity and for, of course, the chemical compounds and the other items we needed in plant tissue culture. Then we need to educate the good technician for our laboratory, for our commercial laboratory. As you can see, about 60 to 80% of the cost is belong to this item. So that shows it's very important having a good technician for the commercial production. I would like at this time to compare two strategies for micropropagation or using the plant tissue culture actually. In India, you know, many commercial companies are active, about 100 commercial laboratories. With different plants that are doing the tissue culture, fruits, like medicinal plant, ornamentals. But as you can see in this slide, Since the first established company in 1986 with the capacity of 1 million plants per year, until now, you can see that the rate of the produced plants is not proportional with the installed capacity. Maybe it's 1/10 of the installed capacity. That showing that concentrating or focusing only on the micropropagation maybe is not a good solution. As you can see here, in the recent years, the number of the commercial plant tissue culture laboratory in India is decreasing. I can say maybe it's because of the wrong strategy or wrong competition. But when we see the strategy in Germany, this is the data for 2004, you can see they are using different technology of the plant tissue culture, not only micropropagation or mass propagation. They are using 23 different applications of plant tissue culture, like in vitro storage, pathogen elimination, anterior culture, embryo culture, somatic embryogenesis, bioreactor, and secondary metabolite formation. So this strategy is very helpful because they can do many different types of the business with in vitro culture, not only micropropagation. And one of the important points, they work on wide area of the plants, like more than 180 different genomes are working. So this is the important items. So I can say in developing countries, we mainly focus on 2 applications of the plant tissue culture, which is like micropropagation, and then maximum like producing a disease-free plant production. But we need to have to establish the companies for, of course, in the research and institutes, in university, they are working different area, but I'm talking about the companies, the startup companies. We need to have some companies in secondary metabolite production, haploid culture for seed production, in vitro mutation breeding, genome editing. As you can see in US, for every $100 million research expenditure, only 1.2 new products been commercialized, and only 1.6 startups are formed. So this is not easy. It's very difficult. I don't want to say we need to expend this much money, but I want to say that it is a very difficult anyway to produce a new product to form one startup company. In Iran also we have the like more than 50 commercial laboratory working in different area of the medicinal plants, very low, but mainly horticulture, like fruit trees, ornamental plants. But the main problem is, as I mentioned, is only focusing on micropropagation. So limited number of the plants. Propagation protocol. The source of the plants mainly usually is not clear. High production price because of the wrong strategy and lack of the R&D team. So for solution for this one, we hold several webinars, several capacity building webinars, and also workshops for clarifying the role of the R&D, production management, variation in production is very important, and also the marketing. For this, solving these issues, we recently established the plant tissue culture accelerators. Until now, we have 6,000 knowledge-based company in different fields, and 500 is biotech company. Of course, main of these biotech company are for pharmaceutical and medicinal purpose, but also in tissue culture. So we try to teach them and try to help them with mentoring, with business support, with seed funding, with finding, especially with finding the investor to try to help them to find their way and not going to the wrong way and not going to choose the wrong strategy. Of course, we already have incubators, but usually incubators are not working very well because they are non-profit usually, and then they don't pay attention that much to the mentoring and business supports. That's why accelerators are important now. So we teach and tell different companies about the variation of production, different techniques, different plans they should choose, like we introduce them like a successful commercial laboratory in the world for different plants and different techniques. We teach them that they should be aware in case of the micropropagation, the method of the subculture, the using the plant derivatives, the items they have to be concerned about that, and also the we introduce them and tell them about the many lawsuit issues recently the companies are facing in Iran. And which method is important for them. For example, for gerbera, washing is one of the big problems because of the many times subculturing or using high concentration of the hormone, or sometimes using TDS-around. And also, for example, danger of the off-type plant production is very critical, and because of the DNA methylation, different subculture and using different high concentration of the hormones, flower abnormalities and bushiness may happen. For example, in case of the date palm, this is a photo of oil palm, but for date palm in Iran, we have one lawsuit issue, which is a big problem nowadays for the producer. Also, application of different plant biotechnology and plant cell culture in cosmetic and food, introducing the new techniques, new companies, different applications, like producing the chocolate from the cells from the like cell culture. Also, some like interesting idea like production of the home bioreactor. This is the cell pod which you can produce the cells in your home and consume it as a food, and which is commercialized in Europe or other countries, and also introduced in many successful companies in plant tissue culture, which they use the cell culture for cosmetic production. And another application could be in vitro mutation breeding. As you can see, there's many applications. For example, in flower, 20% of the all in vitro mutated plants are in flower, and we can use this technology for startup companies. So we try to create the new startup companies in different fields of the plant tissue culture. And of course, we try to teach the companies that work in micropropagation to do it in the right way and to be able to have a competitive price and also high quality plants. Okay, in the last part, I just briefly introduce some ABRI's achievements in plant tissue culture. About 15 years ago, we developed a virus-free plant potatoes. And interestingly, from that, I can say simple technology, now 10 million millituber each year produced. We could save $40 million during past 15 years, establish 25 companies and more than 220 jobs. So now it's very simple technology, I can say, but I just want to emphasize that any technology can have many advantages for the country. Production, saving the money, establishing new companies, new high quality job creation. Recently, we developed a micropropagation protocol for date palm, and we transferred the technology to the commercial company in Iran, and we expect to have a revenue from the export of this date palm, which is called the majul, like about $100 million in 2032. And also we developed plants, virus-free plants for different root stocks. And many companies now established based on this technology in Iran. One of the important topics is hybrid plant production for the F1 hybrid seeds. We developed many technologies for hybrid production in wheat, in canola, in sweet pepper. We already commercialized inbred lines from this inbred lines with, in collaboration with private section. 4 new variety cultivar released by the this technology, microspore emergencies, and also hybrid induction in cucumber. We are also using some new techniques, reverse breeding, with using genetic engineering, so I think of the DMC1 gene and production of the silence plants, and we are going to have find a marker for the choosing the parental to introduce the new hybrid from this technique. Also the new synestry molecular-based method for tomato, also using genetic engineering and genome editing for drought tolerant in rice. Our colleagues could make a very low cost bioreactor until the 60 liters. It's very cheap, and we could succeed to produce many medicinal plant bioreactor like valeriana, as you can see here, and also Withiania somnifera. This is all produced in our low-cost bioreactor, and Echinacea purpura. And finally, I want to say that innovative production can guarantee the future. And I think all developing countries should, and researchers or startups companies should pay attention to this important item, that we should not only copy and just do only the propagation, but of course, if we do the macro propagation, we should do it in the high quality, but we should find a new method in innovative production and system for our job. And finally, I would like to thank you for your attention, and also special thanks to my colleagues, Dr. Malusi, Ibrahimi, Dr. Amirian, Dr. Shayat Hai, and Dr. Masenfo. Thank you so much for your attention.
Thank you very much, Professor. It was very comprehensive and again, excellent presentation. With this, we have some questions in the chat box. If you allow me, can I take some questions for you?
Yeah, sure.
So one question is from Mr. Mehdi. He's asking how activated charcoal is act as a reservoir for the plant production. Number 2, how fungal contamination could be eliminated in a tissue culture lab?
Yeah, in case of activated charcoal, there is many pores inside of the activated charcoal. It can absorb many toxic and phenolic compounds. That's why it's very important, and that's why we need to use high concentration of the hormone. For example, if your protocol is, for example, 1 milligram per liter BA, when you use the activated charcoal, you needed to increase it to 2-3 times at least. In case of the concentration of the activated charcoal, for example, if you use 0.5 gram per liter, maybe 2-3 times is okay, but if you use 3 gram, for example, you need to increase more. And in case of the fungal contamination, you know, the fungal contamination is very important issue for any commercial lab. And the main item is, for example, you need to sterilize well your plants. You need to check first, and then if it's not contaminated, you should transfer to the room your commercial grows soon. You should remove all items which might produce the fungi, like even like high humidity condition. Fungi always is there, and is not like you can make it like zero. You always just need to be careful about different items. For example, in your laminar A4 system always should be checked, or you need to have a like a filterized air inside of your laboratory. Or for example, the window should not be open to the to the yard. It should be closed, a fixed window. All small items should be controlled to be able to control the fungi or bacteria. Otherwise, you will find always the problem, and it's not easy to get rid of the fungi. Yeah.
Okay, thank you. I hope that answers his question. We have another question from Ms. Farhanda. She's asking why we need to do tissue culture of aloe vera, significance of aloe vera tissue culture.
Actually, we don't need to do the tissue culture of aloe vera, because aloe vera is the plant very easy like propagate by the traditional system, no need to do the tissue culture. If you have, in general, if you have any new cultivar with some specification, okay, you can do the tissue culture for aloe vera. Otherwise, then if you want to use the normal aloe vera, you can find everywhere in the greenhouses. You don't need it to do the tissue culture because the production is not economically, and then you cannot sell it actually. You should know the method of the propagation for this plant is no need to be the tissue culture. So if you can find it in the greenhouse production with cheap price, then you don't need to do that. So then my advice is you should know which cultivar of aloe vera. If it's normal one, no need. If it's very new one, yeah, you can do the tissue culture.
Okay, thank you very much. We have another question from Dr. Muhammad Nawaz. He's asking why tissue culture is still not successful technique in sugarcane breeding.
I don't have any experience about the sugarcane, unfortunately. I don't know.
Okay. So here is another question from Mr. Muhammad Imran. What is the reverse process of tissue culture?
What do you mean with the reverse process?
I would request Mr. Imran to explain on it further, but if we can take another question. How much chances of SOA clonal variation in the tissue culture?
This is a big issue for the production of the fruit plants mainly, and also even the cut flowers. Some companies, they thought that because the flower is not going to be used as a not edible, then it's not important. But working with the ornamental plants like pot plants and cut flowers, or horticultural plants like rootstock is very, very different with other plants. For example, usually with doing many times subculturing of any plants, we did some molecular studies where we could find the different genetic differences. After 15 subculture, if you go to the 20 or 25, you can see easily the genetic changes if you do the molecular markers. And or if you use the aggressive hormones like TDZ or 2,4-D in high concentration, you can find big difficulties in your production. No, we have some issues in Iran also. There are some companies that they didn't know the importance of the commercial production. They have to be careful about the production of the true-to type. And the suboconal variation, it could be a big issue. Imagine you are going to sell the rootstock to the orchard man, and after 5 years is going to this orchard man is going to have a fruit or even more, and this orchard is going to be for 40, 50 years. So how you can give them the low material or low quality plants, which is not going to flower, which is not going to have good roots, because in rootstock the root system is very important. So, of course, this is going to cause a problem. Then I just can say, be very careful about the commercial production. You cannot use any hormones, any PGR. You cannot use any like aggressive or high concentration of PGR. You cannot do many times subculturing. After 13, 14 times, you should stop, because more subculture will get chance of the more like variation, mutation, methylation, and then you are going to have abnormal plants. And usually, maybe you cannot see the abnormality in the plants in the greenhouse, but when it goes to the orchard, and after that, you can see the root types has a problem, the whole plant has a problem, or especially in cut flowers, we can find bushiness and they are not going to flower as a normal plants. And of course, imagine the one greenhouse with very high cost of production of the, for example, orchid or anthurium or gerbera, and they cannot get good flowers after 2-3 years. Of course, this is going to be a big problem. Okay.
Okay, thank you. We have another question from Ms. Aisha Noor. She's asking, can 2 callus from tissue culture be combined?
What does it mean? Like 2 callus from tissue culture?
Yes.
I don't understand the question. Actually, I don't know. What is the
Okay.
meaning? About the callus, I can say in commercial propagation, we should not go to the callus production usually. In sometimes very like special cases only. But if they means like protoplast fusion, if the combination means that one, the callus cannot combine, but if you remove the cell wall and then isolate the protoplast, the protoplast fusion can happen here.
Right. So we have another question from Dr. Ahmed Waqas. He's asking, have you tried in vitro regeneration of cotton?
No, actually, maybe in next 2-3 months, we are going to have a project on cotton in Abu, but no experience yet in my case.
Right. So here is another general question from Ms. Sania Shafiq. She's asking how we can use tissue culture technique in a low cost in developing countries.
Yeah, is easy establishing the low cost plant tissue culture. I think is not difficult. In Iran, for example, now many facilities, the big facilities are made in Iran, and we can have the laminar flow autoclave made by Iranian companies. But anyway, for any laboratory, we need to expend some money. For example, we need to have one laminar flow. Or of course, we sometimes, for example, in Thailand, if you visit some laboratory, they use very cheap laminar flow system without filterized like high HEPA filter. That one is another issue, but we need a good autoclave. Usually it's not very expensive, but for commercial use, we need to use the high quality material. Otherwise, you are going to find a problem for your production system. So I can suggest that we should not go to the low cost production. I don't mean we use high or expensive material, no, but we should care about the quality if we want to do the commercial production, because if we produce high quality plants, we can earn enough money without caring about the cost, because it's very I think it's very economically and you can earn good money if you produce the high quality plants. But the most important thing is production management. You should know about the management of the laboratory, for the labor, for reducing the contamination. These are the high costs in each laboratory, not the facilities. You buy only one time, some facilities is finished, but you always need to pay money for each month for the labor. Each month you should pay money if you cannot control the contamination. So you should pay attention to this step, not the cost of the facilities, because you can't find, of course, the good facilities with low price in each country. But this is not the case of the successfulness in the plant tissue culture, I think. Yeah.
Thank you. So here is a last question for you from Mr. Jabbar Ali. He's asking, is tissue culture applicable for apricot and walnuts?
Yeah, in case of apricot or any other like fruit plants, we don't do the tissue culture for the cultivar. We do the tissue culture for the root stock of the cultivar. So that's different. We use it for root stock like GF, GM, or for other plants like for like pre-dwarf, gizella. Tissue culture used for micropropagation of the root stocks. And if we want to use the science for the grafting, we use the mother plant orchard, not the tissue culture. This is for apricot. For the walnut, yeah, this is a very good technique for the walnut. And last 2 years, I visited some companies in Italy, and they told me the plant tissue culture for walnut resulted in very good plant in the orchard. So it is a very good technique for propagation of the walnut. Yeah.
Okay. I think we are having some more questions for you, but there is one question which I think you would like to answer. Can tissue culture cause variation in the chemical constituents of plant?
I didn't get the question. Could you repeat it?
Yes. Can tissue culture cause variation in the chemical constituents of plant?
Okay. Of course it can if you use subculture many times and high concentration of hormone. But if they use the medicinal plant, I think they're talking about the medicinal plant. Usually the medicinal plants, after the micropropagation, you could talk about the chemical compound inside, because always, the medicinal plants have a high compound in the stress condition. So it might have some change in the chemical compound inside of plants, because in plant tissue culture, in vitro culture, you have a very controlled growth chamber. So of course they do not receive that much stress that they have receive in the nature. So that is effective, but I don't know what is the point of this question. Yeah.
Okay. Thank you very much, Dr. Azadi, for your time and wonderful presentation. We hope to see you in Pakistan sometime soon when the COVID situation subsides. And thank you very much again for your time.
Thank you for inviting me. Have a successful webinar.
Thank you very much. Dr. Sama, would you like to say something?
Thank you so much, Brother Azadi. If you have time, please do with us for the remaining workshop. And
sure.
If you allow us, we can share your email with the participants so they can ask more questions if you wish.
Sure. Yeah, no problem.
Thank you so much.
It's my pleasure.
Thank you very much. Now we will move on to our next lecture. This next lecture will be delivered by Dr. Iftikhar Ahmed. Dr. Iftikhar Ahmed is an associate professor at Institute of Horticulture Sciences, University of Agriculture, Pakistan. The title of his talk today is Application of Tissue Culture Techniques for Floriculture. With this, I would request Dr. Iftikhar to please, start his lecture.
Bismillahirrahmanirrahim. Good afternoon, everybody. Hope everybody is enjoying today's course. And I am Dr. Iftikhar Ahmed from Agriculture University, Faisalabad. Currently, I am working as assistant professor, floriculture and landscape. And I would like to thank the organizers of this course for providing the opportunity to talk about the tissue culture of ornamentals. So before moving to the tissue culture, first of all, I would like to share background information about the ornamental horticulture. You might be aware that basically ornamental horticulture is a branch of horticulture which deals with different flowers, including cut flowers as well as loose flowers, and a wide variety of ornamental plants, including landscape designing. All these come under ornamental horticulture, and this business is nowadays worth billions of dollars in global markets. Nowadays, actually, the definition of the horticulture has a little bit changed from the previous one. Now it is said that horticulture is that kind of agriculture which provides food for the body in the form of fruits and vegetables, as well as it provides food for the soul through aesthetic gratification by different ornamentals. So there are a wide diversity of ornamentals which are being grown and traded in the world. But the major component is the cut flowers. So cut flower, what is a cut flower? Basically, any single flower or an inflorescence which is cut along with its stalk or stem. So that is termed as cut flowers, like roses, gladiolus, liliums, tulips. All these are major cut flowers which are being used in the global markets. Then there are some loose flowers like roses, rose petals, which we use for making garlands, which we use for value addition. All these come under loose flowers. Then there is a cut foliage. Basically, there are a wide range of different ornamental foliages, which have also high economic value in the global market. And so those come under cut foliage crops. Then ornamental plant production nowadays, it is getting momentum, particularly with the change in lifestyles of the people, as well as with this CPAC. So there is great demand for the different trees and shrubs, ornamental plants. So their nursery production demand has increased manifold since last few years. So we have a scope here where we can develop our floriculture industry or ornamental horticulture industry on those on scientific lines, and we can fetch maximum return from ornamental plant production. Then for production of those all cut flowers, cut foliages, and ornamental plants, their seed production, their nursery production, and in case of geophytes, their bulb production, that comes under their basically planting material production. Along with these major components, nowadays there are some specialty ornamentals like preparation of bonsais, production of cacti and succulents, and development of strawberries and pseudo bonsais. So they are getting more preference by the people, and nowadays they are in high demand in different parts of the country. So these are just a few major components of ornamental horticulture, and which are I will show in next slides, which are very important in different countries of the world. Although these are very important for Pakistan, but so far we are nowhere in the international trade due to certain reasons we will discuss very briefly. Every year, millions of different ornamentals are being bred, new varieties are being produced, and these are being propagated commercially in order to fulfill the ever-growing aesthetic needs of the people. So still in our local industry, we have limited number of crops and limited quantity of flowers available. But in developed countries, there is huge demand, and we can produce good quality flowers and ornamental plants, and we can export them to different developed countries, and we can earn a lot of foreign exchange for the country. Not only we can earn foreign exchange, but also we can beautify our surroundings as well as our indoor environment, because research has proved that many of the ornamental plants, or rather most of ornamental plants, they mitigate the climatic change, particularly through carbon sequestration, as well as absorption of different pollutants present in the indoors. Last but not least, because it is an intensive kind of production of crops. It demands a lot of manpower, so it produces job opportunities, as well as it is a source of or more income for the not only for the growers but the other stakeholders which are involved in its harvesting, marketing, or export of these ornamentals. So being said that, what is ornamental horticulture, I will just briefly talk about its scenario globally. As we have discussed that these are different floral products which are being marketed in international markets, but here you can see it is around more than 90 billion US dollar industry. So it's a huge industry, and particularly in this production of different plants, their planting material, it has a reasonable share in that total volume. So particularly for production of disease-free cultured plants, tissue cultured plants, and those plants which are not easy to propagate by other vegetative means or through the seed. So for that purpose, their propagation through tissue culture provides us a reasonable opportunity for not only earning more, but also engaging more of our experts as well as youth into this business for production of good quality plants. Here, if you see, although it's a quite 6-7 years back data, but still just it is just to show you that basically in total volume, almost more than 50%, more than half of the production is for the cut flowers. Basically, this is the major segment. And at the same time, around 45% is ornamental plant production. And particularly in this graph, if you see that after roses, tulips, and chrysanthemums, there are the next 4 crops, gerbera, freesia, lilium, and dianthus, these carnation. So basically, these are mostly propagated through the tissue culture. So there is a huge demand, and at the moment, all of these plants are being imported from other countries. So still we are spending a lot of foreign exchange for importing their germplasm. So if we can produce locally, so then we can save that foreign exchange, and we can also provide good quality locally produced tissue cultured plants. So here I will show you a little bit about the floriculture map of the world, how it is being traded. As most of you people will be familiar, that Holland or Netherlands is the leading country in the world, which has almost 43% of total floriculture trade in the world. Almost a couple of decades before, it had about 58% share in the world market. But with, I will show you next slides, that with the movement of or shifting of the production from Europe, from America to Latin America, as well as to the African countries, so which are near to the equator, their local production has decreased, and now they are being those plants are being grown in those countries which have cheaper labor, which have good quality soil suitable for flower growing or ornamental plant production, and then they have good, reasonably favorable policies and environmental conditions. So for that purpose, here I will show you that basically the most important thing is that Netherlands have only area under floriculture production, which is about 20,000 acres. So it is even less than the area still we have in Pakistan. But the thing is that they are sharing 43% of the market, the value. But if we talk about our country, we are nowhere on this floriculture map. Their major crops, most of the ornamental crops are being produced. And basically, this is a typical example of Netherlands. So how they are capturing the whole floriculture market of the world. Basically, first of all, they produce germplasm. They produce saplings, they produce seeds, they produce bulbs. They export these planting material to those countries where there are comparatively cheap resources and favorable climatic conditions. And then through corporate farming, those crops or those plants are being raised in those areas, in those countries. After raising according to those standards, then they import back. And after importing, they brand them. And then after branding, they re-export them to different European countries, America, Japan, Russia. So big countries, the developed countries where there is high demand and there is more tradition of using flowers and ornamental plants in their daily life. And basically, all this is done through innovation, through advancement, and through their branding. So if we talk about next, I have told you that before a couple of decades, most of the production was in the Europe and America, but because there was comparatively expensive labor, so they had shifted to those areas, and even cost of production was high due to the unfavorable climatic conditions during some part of the year. So then they moved their floriculture production into those areas or those countries which are near to equator. They had same climatic conditions most of the time throughout the year, and they had good quality soils, and they had cheaper labor and other resources, and good government policies to support their floriculture industry. So in Latin America, Colombia is the leading flower and ornamental plant producer, followed by Ecuador. Similarly, in Kenya and Ethiopia. So these are in Africa, and there is a fifth country which is emerging, that is Malaysia. So these five are the countries which are located near the equator, and these are now these are major these have become major players in the floriculture market. So all these five countries have area under floriculture production that is almost near to the similar as Netherlands. So 20,000 acres approximately they have area. They are growing most of the major flower crops as well as ornamental plants. But and you can see their market share is even 1% higher than the Netherlands. So these five countries are holding almost same marketplace as the Netherlands. So what are their competitive advantages due to the favorable climatic conditions for most of the time of the year? Their lower cost of production and cheap logistics, as well as their the government policies for good quality production and supporting the floriculture industry. If we talk about China, in the world, China has maximum area under floriculture production. So almost 200,000 acres of the area is under floriculture production in China. But if you see, they have only 2% market share. What is the reason? So basically, they have their huge local market. So most of their production is being utilized locally. So they are number one leading country in the world which have highest area and highest production of ornamentals. And even some of the tissue cultured plants are being imported from China to Pakistan, although since last couple of years, government has put a ban on importing some of the plants. So basically, again, their competitive advantages are favorable climatic conditions in some parts of the country, their lower cost of production. As we know that most of the Chinese products, they have comparatively low cost of production, and their easy market access and government support. So after this, India is our neighboring country, and I have added this one just because of the reason that in tissue culture ornamental plant production, India is the leading country. They have around 81 different companies which are producing tissue cultured plants, and they are exporting to different countries in the world. Along with that, after China, India has the second largest area under floriculture and ornamental plant production, which is about 170,000 acres. And they are producing a lot of different crops, flowers, as well as tissue cultured plants, as well as loose flowers. But still, its market share is also 2% because most of the production is local production, and that is being utilized in the religious ceremonies. Basically, cost of production is also lower, climate is also favorable. So we have also similar type of competitive advantages in our country, except some lack of infrastructure and particularly government or public sector support for promoting this floriculture industry. Iran is emerging. Their area is also similar, which we have, but still they have very nominal, just 0.065% share in the world market. These are all the major importers, particularly USA, Germany, Belgium. Most of the flower and ornamental plant consumption, per capita consumption, is highest in Europe. European people have more buying, purchasing power, and particularly they have tradition of buying more ornamental plants and flowers for their daily life. If we talk about South Africa and Australia, they have small floriculture industries, reasonably small, but basically these are specialty floriculture industries because these work with the native flora. And this is another segment where we should work, that we should explore our native flora in a way that we can grow our native flowers and we can market. This is what Australia and South Africa is doing, and they are earning a lot of foreign exchange for their countries. And rest of the all world has almost 300,000 area under floriculture, and all of these countries contribute only 8%. And here you can see just Holland has 43%, 5 countries, Colombia, Ecuador, Ethiopia, Kenya, and Malaysia, they have 44%, and remaining just 2% are the rest of the world has 8%. So due to certain niche and different regional advantages. So here I think it will be more clear to you people that how floriculture is going all around the world. So after being said about the world, then I will talk about a little bit about Pakistan. So currently in Pakistan, we have area of about 23,000 acres under floriculture production. Of total arable land, it is just 0.5 or 0.6%. So here you can see even we have higher area than as compared with the Netherlands. But we have almost negligible share, which is just 0.0255%, and that is only from dried rose petals. We are exporting to Gulf countries. Although we have some flowers like roses, tube roses, gladiolus, marigolds, but since my postdoc back in 2014 afterwards, we have started working on diversification of floriculture, and we have introduced some new flowers which are being now grown as specialty cut flowers commercially. So, and these are also available in different major markets of Pakistan. We will talk about that later because we need to focus on our actual topic. So just to have a background knowledge about ornamental horticulture, so I thought that a little bit introduction of ornamental horticulture would be of interest for the participants. So after this, here if you can see, this is just the graphical presentation of that one. Okay, here you can see all the 5 countries. These 5 countries contribute 44%, Netherlands is 43%, and rest of the world is 8%. So if we talk about the global scenario of tissue culture of ornamentals, as per information available in different reports, currently over 500 million plants are being tissue cultured every year, and majority of these plants includes ornamentals. So ornamentals have high value because most of these are high value. Theoretically, we can, as my other presenters have mentioned, that theoretically we can go for tissue culture of any plant part or any plant, but it is only beneficial for those plants which have high value. As many of the ornamentals are high value crops, so we can go for production of high value crops through the tissue culture. Among different world countries, 81 countries are basically importing different tissue culture plants, while 33 countries are leading exporters for the tissue cultured ornamentals. And these exporters include India. As I have told you that India is leading in tissue cultured plant production, and they are mainly exporting this to Australia, Japan, as well as to European countries. Then Netherlands, because Netherlands also export different planting materials than Vietnam, USA, China, Colombia, and Italy, as well as Israel. So they constitute almost 80% of the world market. So these 6, 7 countries, they are providing 80% of the tissue culture plants to the global markets. Among importers, again, Netherlands, because Netherlands also, because as I told you that they several times export and import and then re-export just after branding different products. So Netherlands, Australia, USA, Canada, Mauritius, Japan, and Kenya, they are all leading importers because they import tissue culture plants, they grow them, and then they export them. Among crops, according to a report, almost 156 ornamental genera are being propagated through tissue culture in various laboratories in the world. While among these, gerbera is the leading crop because it's a famous cut flower and as well as potted plant, mostly for cut flower production. Gerbera is propagated through tissue culture and followed by carnation, orchids, philodendrons, spathiphyllum, peace lily, then dracaenas, Diphyndigia, cordyline, monstera, pyxis. All these have high market demand in global market. There are many other crops, but these are some of the potential and leading crops which have high demand in the global markets. And there are over 750 companies in different countries which are doing this tissue culture plants business, and 236 companies with only 81 countries only 81 companies in only in India. So they are marketing or producing as well as for local market as well as for the export to the different world countries. So after global scenario, if we talk about local scenario, so just it will make it clear that where we stand regarding this, particularly for ornamental plant production. So here in this one, although there are more than 2,000 ornamental plant nurseries in Pakistan, but unfortunately, still so far, there is no commercial tissue culture lab functional for only for ornamental plants in Pakistan. So this is major dilemma that we have to import tissue culture plants at plug stage, at nursery stage from different countries like China, Thailand, and India. There is a huge demand. Many people, they are interested to buy and they need to import these tissue culture plants. But unfortunately, still, although we have started working on that one, but commercially, nobody is doing that. So we need to focus on that one. If we can establish commercial tissue culture labs, particularly at Patoki. Patoki in Kasur is the hub of floriculture trade in Pakistan. So they several times they have asked that they need trained personnel for tissue culture propagation of the different plants with. And if we can have some such type of setup with public-private partnership, so that would work best for the uplifting of this sector. And we also need to develop specific protocols under local conditions for different commercially ornamental plants. Currently, we have more demand for gerbera, carnation, and lilium, with some demand in orchids. Orchids are not so common here in Punjab, but for Karachi particularly, it is one of the best crop. And I knew that in past, HEJ Karachi, Rao Saab was working on that, but I'm not sure what is the status of tissue culturing orchids in Karachi nowadays. If we talk about different ornamentals, almost you can propagate any type of ornamental plant, but the crops which have high demand, I have just listed those ones. Among cut flowers, we can propagate roses, carnations, gerbera. Gerbera is leading. It has highest demand, not only in Pakistan, but all over the world. Then lilium, dahlias, orchids, etc. Then if we talk about potted plants, dracaenas, araucaria, Diphyndigia, these are basically beautiful foliage potted plants. And there are several others. Then indoor plants, calathea, Haworthia and Aglaonema, we can propagate through tissue culture in trees. Sagwan nowadays, it is getting popularity. Then Alstonia, particularly because to produce disease-free plants. Nowadays, Alstonia has a gall problem, so we can control that problem by propagating it through tissue culture, Terminalia, and many others. In shrubs, spices have high demand, although these can be easily propagated through their cuttings. But actually, you can plant cuttings only in 2 seasons. But if you want to produce throughout the season or throughout the year, so then you may go for tissue culture, mass propagation. Then Legistomia, fruits and vegetables are not our topic, so we will skip that one. There are many other crops, but these are some of the potential crops which need to be tissue cultured and which are in high demand in local markets as well as in the global markets. These are all almost same crops which I have discussed just to show that these are different ornamentals which have high demand in different markets. And by just producing them, particularly for the ladies, it is a very good enterprise for women empowerment. If we can train our young generation for tissue culture propagation, so that would be worth lowering our import bills. So having said that, now we will move to plant tissue culture and we will take the case study for gerbera. And before that, as already the presenters have mentioned that what is tissue culture. So basically, we will not go into detail, but briefly, in vitro and aseptic cultivation of any plant part just to have mass propagation, and you can use any plant part for that purpose. So that is plant tissue culture. These are some basic terms. I think everybody is familiar, and that has been said. Explant are those parts for just for the students that any plant part which is being used for culturing on growth medium, that is explant, and the medium which is liquid or semi-liquid on which that explant is being fixed, that is growth medium. Transfer of that explant into the that growth medium, that is called inoculation. And after inoculation, then you need to incubate for a couple of weeks or a few weeks at a specific humidity, temperature, and light intensity as well as light period. That duration is called incubation period. Finally, callus, mass of undifferentiated parenchyma cells, and sterilization is very important in tissue culture. You need to have everything sterilized. You need to remove all sorts of germs in order to have successful tissue culture plants. So basically, the tissue culture is based on theory of totipotency, so which we will discuss that it is the ability of each and every cell to produce a new plant. Similarly, another principle of tissue culture is plasticity, which is the ability of the plant to change their growth and development according to the environment in which they are being grown. So from cells, from single cell, then tissue develops, and from the tissues, organs develop. After the from organs, then organ systems, and finally organism is being produced. And when there are a lot of organisms, there is a population or mass of the mass propagation. So question arises why we should go for tissue culture. I think this has already been told, so that's why just briefly why we do tissue culture in ornamentals. Most important thing is that to produce mass number of plants throughout the year with no limitation of the season, because most of the ornamentals, these are being propagated in particular seasons if we are doing by other means. But if we are going for tissue culture, so we can produce almost year round and we can produce mass number of plants, millions of plants in less time and in less space. Then second most important benefit of tissue culture is disease-free or virus-free plant production. There are many plants which are badly affected by the different viruses, and if we can use different techniques of tissue culture for producing virus-free plants, so then it is the best way to produce those plants. so we can get identical clones, and it is most importantly best for those plants which cannot be propagated by other means. We can do breeding, we can go for disease-free plant production, and we can also use micro budding and micro grafting. These are different techniques which you can go at the industry, at the nursery stage for propagation. These are some other benefits. So mainly, I have told you that basically for crop improvement and production of varieties which are resistant to different biotic and abiotic stresses and the viruses, so we can go for tissue culture plant production. There are different techniques of tissue culture which can be used for propagation of ornamentals. These include cell culture, callus culture, embryo culture, seed culture, meristem culture, which is most commonly used in ornamental plants, then protoplast culture, organ culture, and pollen culture. So in ornamental plants, we can use most of these methods and techniques, but commonly meristem culture is more commonly used, or callus culture. So these techniques are commonly used for raising of different ornamental plants. So these are some further techniques which we can use if particularly we are going to discuss, we are going to produce new varieties, or we are growing to produce some disease-resistant plants. So we can use any one of these techniques for raising of different ornamental plants. As these techniques have been discussed by my previous presenter, so that's why I will not go into detail, and we will move to different steps, particularly which are required for the tissue culture of gebra. So these are different steps or stages of involved in the tissue culture of ornamentals. First of all, you need to prepare the explants. Which type of explant is best suited? We have conducted some trials. I will show you in next slides. So after preparation of explant, then we need to prepare the growth media. So in which media we need to grow that explant. Then we need to transfer that explant. After sterilization, we need to transfer that explant into that media, which is inoculation. And finally, after inoculation, we need to incubate that one in the growth rooms for a few weeks. Now, next comes multiplication. If you are you need to produce more plants, you can use that newly produced plant, and that stage is called multiplication. You can go back to the inoculation stage, and from inoculation, then incubation, and finally, when you have produced your required quantity of plants, then comes hardening off. So we will discuss how we can harden, because we have produced plants in the lab, which is totally under controlled conditions, and we need to shift the plants in the field. So if we will directly shift them, so they will not grow. So that's why, and they may wilt. So that's why we need to first make them harden, and so that they should be capable of withstanding the stresses in the field, and then we should go for transplanting in the field. So in tissue culture, different substrate, different mediums are being used. So basically, the most common medium, as you are familiar, that Murashid and School is very famously known as MS medium, is being used for tissue culture of different plants, which includes macronutrients, micronutrients, vitamins, iron, sugar, and then there are different growth regulators which are being used, and particularly agar is being used for solidifying the medium. So one by one, if we'll talk about the preparation tissue culture, first of all, the preparation of explant, how we should select explant. First of all, you need to select healthy, vigorously growing plants, and the branches should not be too young, and those should not be too old. And these should be free from any insect, pest, or disease, or any type of wound. And if you can grow that plant under controlled conditions for a couple of weeks, and you can, that would be better. And then you need to always use a sharp blade or sketcher to cut that explant from the parent plant. When you have taken the explant, then most important next step is sterilization. So you need to sterilize the explants. So there is a complete method. First of all, you need to wash with the tap water for a couple of minutes, and then you may need to put in the 70% ethanol solution for a few minutes, and finally you need to wash with the distilled water. At the end, you can use bleach solution, 5 to 10% bleach solution, and a few drops of Queen 20 for rinsing that one. And at the end, finally, again, you need to rinse that one with the double distilled water for some times, so that it should be all type of micro free. It should be all type of chemical or detergent free. So this is very important step, and it should be done properly so that you can have less problems of contamination. Then afterwards, you can resize them. Generally, if it is required, you can resize. Otherwise, there is no need to resize if you have already taken small sizes. But according to your requirement, you can, after drying them, you can resize them in the inoculation room in air laminar flow hood. Size of the explant should be around 2 to 3 or 4 millimeter. So that would be fine for propagation. Then next step is preparation of growth media. As I have told you that there are different components. You can make stock solutions, and finally, after making stock solutions, then you can adjust the pH, which should be around 5.8. And afterwards, then you can add sugar and agar. And finally, in this way, you can make the volume. And then after sterilizing the glass sphere, in which even petri dishes or the test tubes which you are going to use, or any jars, so then you can pour that medium into these containers. And finally, after that, you need to sterilize that one also. But one thing which is very important, during tissue culture process, we also use certain plant growth regulators, particularly some axons and cytokinins for shoot and root regeneration. So one thing which is very important, that these are thermoliable. That means these are high temperature sensitive. So these should not be added before sterilization. When you have sterilized the medium, after that you can add those PGRs, and then you can go for inoculation. I have talked about sterilization. When you need to sterilize the media in autoclave, you can fix the time, temperature of 121 degrees Celsius with 15 psi pressure for 15 to 20 minutes for medium as well as for the glassware. After sterilization, then you need to add PGRs, and after adding PGRs, then you need to shift them in the inoculation room. Next step is the inoculation. So basically, it is the transfer of the explants from your transfer explants into the nutrient medium or growth medium which you are using. So basically, it is being done in the air laminar flow hood as shown in the photograph in the slide. So, and again, you need to have proper sanitation yourself, as well as the sketcher and the other helping material which you are using, that should be properly disinfected. And after that, you should go for inoculation, because it is the one of the most important steps when there are more chances of some contamination. So for that purpose, during this process, you need to be careful, and you need to use different forceps, that those should also be autoclaved, and use 90% ethanol just after each use of these forceps or any other material. After you have successfully inoculated all the explants into those test tubes, then you need to inoculate, you need to incubate these all inoculated cultures in such an aseptic growth room where you need to set the temperature for 24 to 26 degrees Celsius with more than 50% relative humidity. And generally, 8 hours of dark is being provided with 16 hours of light. Uh, so keep an eye on any contamination so that you need to remove well in time. And when you have incubated and there is you need to further subculture according to your requirement, then you can go for multiplication or you can go for hardening off. If you need to multiply, it involves calogenesis, shoot regeneration, root regeneration. According to that, you can decide what is your next step. At the end, there is transplanting, but before transplanting, you need to harden your crops. So, because then you need to transplant, you can do that transplanting at the end of the incubation, and you need to shift all those plants into medium. There are two steps. In some plants, you start hardening in the growth room just by increasing light intensity and eliminating or reducing some of the food available to the plants. Whereas in some crops, just you need to take out the plants after they have incubated. You need to wash them under tap water so that all the medium and agar as well as PGRs all are being removed, and then you can carefully plant in small pots having a well pasteurized and sterilized potting substrate. We have developed our own UF grow in our lab, and we are using that one, and that works best for this purpose. You can use any other imported or local medium available for this purpose. After shifting, then you can keep them for a couple of weeks in the same growth room with just increasing a little bit the environmental conditions, light intensity, as well as a light increase in temperature. So it will help in hardening off. So basically, when you need to harden off, this is very important. First, you do it for 1 to 2 weeks in the growth room. Then you shift into the greenhouse. And from the greenhouse, after a couple of weeks, then you can shift them into the field. So this is the whole procedure by which you can do tissue culture of different ornamentals. Here, if you can see from X plant, this is for the basically for the gerbera. You can take shoot ipses after surface sterilization. You can wash them. Then you can go for shoot regeneration. Then you can incubate different cultures. Then shoot initiation. Then you can go for multiplication. Then you can go for rooting. And finally, it is hardening off. And in this way, you can produce different plants. Although it seems quite easy, but it is not that easy step, because many of our labs in Pakistan, they are producing plants. Most of the plants are being produced through tissue culture, but major mortality occurs during the hardening off and transplanting stage. So these need to be optimized for each and every crop, particularly for having successfully tissue cultured plant production. So in this production of different tissue culture plants, there are different problems associated, or there are some certain limitations. The most important is one, the contamination. There may be browning, there may be some exudes of different phenolics. So any type of contamination, or even sometimes if the tissue culture person who is doing tissue culture, that is careless. So due to that negligence, there may be contamination and wastage of all the resources. So when you have limited resources, when you have limited number of explants, so need to be more careful. So then the second one is most important, which I have told you that most important problem here, what we are facing, that is dying during acclimatization or hardening off. Plants are being produced in the lab, but when we are shifting into the greenhouse and then from field, then they go. So that's why we need to work on these. Still, we need to work on this aspect to lower that mortality percentage and to increase the success percentage of the tissue cultured plants. Although everybody cannot go for tissue culture plant production because it's quite expensive to set up a government lab, a tissue culture lab. For that purpose, I think that government should provide some subsidy to the interested people who are interested to start this business, because it involves a lot of different machines like air laminar flow hood and autoclaves and many other materials, chemicals which are needed. So for that purpose, not only everybody can go for, and similarly, not every plant can be or should be propagated through tissue culture. Also, another issue is unavailability of skilled manpower, because many of the people who are working in floriculture sector, they don't have training on tissue culture, so that's why we are having that problem. And fourth one is every species has different protocol, so we need to optimize. Although for 3-4 crops we are working, which are major crops, we are working on gerbera, we are working on carnations, we are working on lilium. So we are developing their optimal protocols so that we can give to the or commercialize those protocols for commercial production of these plants. Because lilium, tulips, freesia, all these are bulbous crops, geophytes, and all of their bulbs are being imported from the Netherlands to the Pakistan, and these are quite expensive. If we can produce, and we are working on that, and if we can produce them locally, so we can save a lot of foreign exchange. So it is but for that purpose, we need to develop their protocol, optimal protocol to be followed for their tissue culture plant production. Another issue is that continuous electricity, which is still, even nowadays, we are having that problem that unfortunately 24 hours electricity is not available, and there may be disruption, and that if that will persist for some longer period, so ultimately that may have issue in tissue culture plant production. And last but not least, as we have many woody plants in ornamental plants, so these are quite difficult to propagate by tissue culture. So then for those purpose, we have to go for propagating them by other means. So this is the same step which I have elaborated, that from parent plant, first of all, you need to put them in the lab for a certain period of time. You need to disinfect them. And then after that, you can select X plants from the mother plant, prepare medium, go for inoculation. After inoculation, then go for incubation. After that, there may be callus. There will be callus induction. From callus, there will be shoot induction, shoot regeneration, and from shoots, you can transfer to a new medium where you can add auxins for root regeneration. Finally, after you have produced your required number of plants, then you can shift them in the greenhouse for hardening, and in this way you can produce your tissue cultured plants. At the end, I would like to share some of the crops results which we have so far conducted at our lab here in Institute of Horticultural Sciences. We have worked on gerbera, and among different X plants which we have tested, petioles, callus, and microshoots, these all three are suitable for their propagation. And different, most of these we have used mainly Eurasian and Stugan MS medium, and we have added different PGRs like 2,4-D, benzyl adenine, and naphthalene acetic acid for root induction, shoot induction, and for regenerative callus. So these, this is their recommended levels, which we have found better as compared with our other treatments and documented in literature. Then dendrobium orchids study was conducted on that one, and these were our results for callus induction, shoot induction, and root induction. So which type of explant was best, and which type of medium and PGRs we found these are optimal, while the response for that what purpose. So then Ixora, for Ixora, we have used these 4 types of explants with the different medium. Mainly benzyl adenine worked better for shoot multiplication or shoot regeneration. For root regeneration, naphthalinic acid was better for Ixora, while for the roses, we have developed a new strain of Rosa centifolia, which produces flowers throughout the year. So we have a huge demand for its saplings. So we conducted a study in which we used its axillary buds, calli, and microshoots, and then with DA and IBA. So these have also proved better for their tissue culture plant production. So we are also working on lilium, carnation. This year we are working on those crops also. So hopefully we will be able to find a little bit protocols, the medium and explants for their tissue culture plant production. But still we are having problems in its hardening up and transplanting, which we need to optimize and which we need to control to lower the mortality percentage, which is comparatively higher so far. All right, so that's all about ornamental tissue culture. So at the end, what are the future prospects? Again, I will say, as I have told you earlier, that we need to have a tissue culture lab, particularly at Pattogi, which is the hub, and they people are looking for some experts in tissue culture for setting up their private labs and for propagation of gerbera and other ornamentals for not only for local use as well as for export. And they are willing to even offer job opportunities if somebody has skills right from start to the transplanting stage. Secondly, in order to ensure mass propagation throughout the year, we need to have tissue culture labs. And most important, there are many of our native crops, native ornamentals, which are nowadays endangered. So we need to save them. And this is one of the best way that we can save our endangered ornamental species. And in this way, we can also help to lower our import bill, which is being spent right now on import of tissue cultured gerbera and tissue cultured other ornamental plants. So I hope by this we can be able to help our country floriculture industry by producing good quality ornamental plants through tissue culture and lowering our saving our foreign exchange by lowering the import bill. So that's all from my side about tissue culture of ornamentals. If you have any question, you are free to ask. Thank you very much for your patience. And if you don't have time, here are some of the information regarding if you need to contact with me later, you can contact through phone, you can contact through email, which is being shown on this slide on Facebook. We have a page. Similarly, recently we have started our YouTube channel just to impart training regarding ornamental horticulture and regarding floriculture crops to the people of Pakistan. Thank you very much. Have a wonderful day and feel free to ask any question if you have. Thank you.
Thank you very much, Dr. Iftihar. It was a very nice and effective presentation, I would say. You can see many comments and questions in the chat box for you. Sir, if you kindly agree, can I take some questions for you? Dr. Iftihar, can I take questions for you?
Yes, sure.
So we have one question from Ms. Farkhanda. She's asking, kindly explain microbudding.
So basically, microbudding is the micrografting technique actually, and it is more commonly used in citrus, but it is a synonym which is used for micrografting. So in which we produce microplants for raising the pseudotype plants for just growing their time stock. So it is not so common in ornamental plants.
Right.
But just for the
Right.
information I had shared.
All right. We have another question from Mr. Asogba. He's asking, why should we add growth regulators after sterilization?
Because most of the natural growth regulators, these are thermoliable. So to increase their efficacy, so we should add them after their sterilization.
Right. Here is another question from Ms. Aisha Noor. She's asking, which ornamental plants are required for the large-scale production by tissue culture technique in vitro?
I think I had added 2 slides, and I have explained all those which
Yes.
have high value in global markets as well as in local market. But as I have presented about gerbera as a case study, so gerbera is the leading crop, but there are many other crops, and even particularly the objective is to produce mass propagation throughout the year, because in normally most of the ornamental plants, these are being propagated in 2 major seasons during the year. But in this case, we can produce crops, the plant saplings all over the year. So in this way, we can produce mass propagation, more number of plants continually throughout the year. And secondly, if particularly we have some problem with some crops, then we can go for disease-free or virus-free plant production. Thirdly, I have stated that basically, although theoretically we can propagate any type of plant, but actually those plants which have high value, and many of the ornamental plants, particularly the foliage plants, the cut flowers, so they have high value in global market. So that's why we should go for propagation through tissue culture.
Thank you very much. We have another question from Mr. Jahazeb. He's asking, sir, can you please explain that whenever we practice nodal culture of crops like guava, they start releasing phenolics and browning can be observed after a few seconds?
Yeah, it's a major issue in tissue culture, but actually if we can manage the concentration of activated charcoal, so we can manage that one. Secondly, the second option is just by subculturing. So these are 2 techniques by which we can lower this issue. Next question, please.
Yeah, we have next question that which ornamental plants are available for the large scale production in vitro?
I have told.
You already responded that. Yes, you
Yeah,
already responded that. Let me check if we have other questions.
There was a question,
Mercurous
rather
chloride.
comment.
There is another one that mercurous chloride is banned in world. What could be the best alternate to that as disinfectant?
We can use sodium oxychloride. So that is comparatively better as compared with that one. Secondly, we can use silver nitrate.
Yeah. The next one is, can you kindly tell names of lilium variety on which you are trying to develop protocol?
Basically, we are Brunello variety. We are working on that one. Lilium is that kind of bulbous crop which is having increasing popularity in Pakistan during last few years. And its bulbs are quite expensive, more than 120 rupees per bulb on wholesale price, not on retail price. So we are trying to develop protocols for producing those lilium propagules from their propagules, and we are trying to produce their bulbs through tissue culture also. So that's why we are working on LA hybrids and Asiatics. We are not focusing on Orientals.
Okay, thank you so much, Dr. Iftikhar. The last question is, what is the reason behind plant not grown or breach out, and sometimes fungal contamination, although try best to follow the protocol?
Basically, fungal contamination is due to the contaminated explants. If your explant is not properly sterilized and it is contaminated, already before when you are culturing, so then there will be more chances of that fungal problem. Secondly, if your staff is not taking precautions, so then there may be the chances. Otherwise, in medium, it is not so common issue.
Okay, thank you so much, Dr. Iftikhar. Please, as you have already mentioned during your presentation, so we will drop your email ID in our chat box also. So if
Yeah, sure,
all
sure.
our participants need to contact in person, they will do that.
You're
Thank you.
most welcome. Thank you very much for inviting me and sharing some of the issues and some of the possibilities and future prospects for improving tissue culture in Pakistan. We are working on that, and we appreciate your efforts for this purpose. Thank you. Have a good day.
Thank you so much.
Thank you very much, Dr. Iftihar, for your time, and it was a wonderful presentation. With this, we will move on to our next speaker. Our next speaker is Dr. Muhammad Usman Rana. Dr. Usman Rana is an associate professor, and he has a postdoc in molecular physiology. The area of his research interest include plant molecular biology, chloroplast transformation tools for the crop improvement. The title of his talk today is Ethics and Biosafety in the Plant Biotechnology. With this, I would request Dr. Muhammad Usman Rana to please start his lecture.
Thank you very much for your introduction. Okay, so I'll be discussing about safety and biotech in plant biotechnology. So before starting the presentation, I would like to give a brief outline that I have made 3 or 4 components, in which, first of all, I would like to discuss pros and cons of plant biotechnology, particularly for students. Then what are safety and bioethical issues? And then the transgene containment, that is the major issue. Transgene containment strategies and technologies, and what is the way forward. So in plant biotechnology, basically we are utilizing the biological principles and engineering principles for the betterment of human life. Plant biotechnology is an umbrella term, so that involves many fields. So so if we look at plant biotechnology, so there are 4 main areas, starting with the genomics, in which we go for molecular breeding, and that could give rise to productivity improvement. Many transgenic crops have been developed, and I will give you details of productivity in the next slides. Then the rapid gain in selection. So transgenic crops have been produced for nutritional improvement, for stress tolerance, including biotic and abiotic stresses. Genome editing, that is very popular field nowadays, involving functional genomics, and site-directed mutagenesis could be done, and mutants could be developed, which could be selected later on. And then molecular farming, in which we can develop antibodies, vaccine production, and even edible vaccine is a very popular field. So there is a secondary metabiotic production, which particularly involves plant tissue culture. We can develop cell suspension cultures, and we could produce metabolites. So collectively, biotechnology is contributing for the elimination of hunger, for malnutrition, for improving the human health, and improving the environment. So the major tools in biotechnology, starting with tissue culture. So since this morning, much has been talked about tissue culture. I will not go in detail. Then genomic analysis. So that involved other omic approaches as well, transcriptomics, genomics, and metabolomics. Then metabolomics is also functional. And the last one is gene manipulation or genome manipulation using transgenics and genome editing. So these are the major areas in plant biotechnology. So first of all, I will start with the plant genetic transformation approaches. So whatever the way you are going to use, first of all, you have to develop a biologic asset in which you involve a promoter at the start of gene of interest, then the transgene that has been taken from some other species or the terminator. Similarly, you have to add a marker gene. That marker could be antibiotic resistance gene, or it could be a visual marker. So many visual markers are also available. So that is your biologic cassette. So you add this cassette into the TI plasmid, that is tumor-inducing plasmid, and you could use the different methods, electroporation, polyethylene glycol induced, or nanoparticles. So that TI plasmid is transferred to the transformed to the protoplast, and then you can develop the callus using agrobacterium. Basically, this TI plasmid, this is transformed in the agrobacterium. So that agrobacterium infects that is co-cultivated on the tissue culture plates with the media, having antibiotics for selection of the desired cells which have your marker gene. Then you go for selection, callus. There are several selection cycles. So basically, shotgun is used, or this is helium gas is involved in the ballistic transformation. So you could use the gold particles, you could use the nanoparticles. So by using these particles, you could coat your DNA of having this cassette, and that could be shot on the ampules on the any plant part which you are using. For example, you could use a deep disc, you could use callus, embryonic axis. So there is another method which is known as chloroplast transformation, in which we are not using TI plasmid, we are using plastid vectors. So in which again in the cassette we have to use the left and right flanking regions. So this plastid vector is that gives you a site-directed transfer of the gene into the host plant through the homologous recombination. So then you go, develop heteroplasmic cells having few plastids that have been transformed. Then you have to go for several selection cycles, and then you develop homoplasmic cells, and then regeneration, and then you could select the transgenic plants. So these are, generally speaking, these are different methods which are normally used for making transgenic plants. So overview of the plant GMOs commercialization. The first transgenic plant, these were basically antibiotic resistant plants which were developed in tobacco and petunia in 1983. Then transgenic tomato that got very popular, flavor saver. This was developed in 1994. Then insect resistant BT crops. This is one of the most popular traits that have been modified. And glyphosate resistant soybean has been developed. That was developed in 1997. So far, 525 transgenic events have been done, and in 32 crops. So these have been commercialized mainly in maize, cotton, potato, canola. So these are the main crops. So transgenic cultivation have given a 113-fold increase, and total 192 million hectare area is under transgenics now. So this makes about 18.2 billion US dollar industry so in the world. So then there are common targets for genetic modification in crop plants. So these involve there are a lot of traits, but mainly insect resistance is the major issue in agriculture. Bioenergy, herbicide tolerance, herbicide resistance, salt tolerance, drought tolerance. These are because of climate change. These are very important traits nowadays. We are interested to enhance the nitrogen use efficiency, water use efficiency because of water shortage. Photosynthesis need to be improved. Biofarming, biofortification, metabolic engineering, biofuel production, that is very important area to reduce the carbon dioxide production by the use of fossil fuels. So these are the main areas, these are the common target traits or the areas which have been focused for the development of genetically modified plants. Okay, impact of biotechnology and commercialized GM crops. So after the utilization of GM crops, their pesticide use has been reduced 37%. Yield has been increased up to 22%, and farmer income has been also been increased up to 68%, particularly small farmers. So these are the commercialized GM crops having mainly insect resistance, herbicide tolerance. In insect resistance, mainly maize, cotton, potato. In herbicide tolerance, again maize and canola and soybean. These are main crops. Abiotic stress tolerance, gain corn, nutritional improvement, and disease resistance. So these are certain main areas where biotechnologists they are working and they are producing transgenic plants, and these plants have been commercialized so far. The GM food safety assessment records by commodity. So if you look at the commodity, main records have been submitted in corn, cotton, soybean, potatoes. So these are the main crops. And if you look at the traits, mainly glyphosate and glyphosate tolerance, then Lepidoptera resistance, against insects, herbicide tolerance, Coleoptera and kanamycin resistance. So these are the main traits for which records have been submitted so far. So biotech crop countries, there are about 18 mega countries which are producing which are growing more than 50,000 hectares of the area, including US, Canada, Spain. Pakistan is also one of the main countries. We are focused on cotton. BT cotton is being cultivated, and then Philippines, India, Myanmar. So these are the main countries. So they are producing different crops. So if you see, there are major number of crops in USA, and Australia is only in canola and cotton. Canada is also cultivating canola, maize, soybean. So these are the main countries. So these are the GMOs which are cultivated in traits of interest in USA. So that is some data about US. So more than 90% is herbicide tolerance, in different crops, cotton, sugar beet, soybean, and then there is virus resistance in papaya. So if you look at the data about global distribution and cultivation and the adoption rate, so the developing countries, the area under cultivation in developing countries of transgenics, that is increasing, that is more than the industrial countries compared to the, and this is increasing every year. If you look at the crops, which particular crop has more adoption rate? So in biotech, soybean that has more adoption rate compared to maize, canola, and cotton and other crops. So coming to Pakistan, GM crop events which have been approved so far. So we are growing 3 million hectare area under GMOs, and mainly there are two events which have been commercialized so far in cotton and maize, mainly BT cotton and Roundup Ready, that is for herbicide tolerance. Overall, that biotechnology has contributed in increasing crop productivity and providing a better environment by reducing the application of pesticides, conserving the biodiversity, reducing carbon dioxide emission by using the biofuel, and helping alleviating in poverty and hunger in more than 65 million people. So that tells us that biotechnology has contributed a lot, and GMOs have enhanced productivity. We have better resistance against biotic and abiotic stresses. Use of insecticide and pesticide is being reduced. Reduction in the environmental containment. More utilization of land resources that reducing by particularly in countries like Pakistan, we are developing new colonies every year. Enhanced nutritive value of the food, particularly good example is golden rice. Long shelf life of the fruits and vegetables. Shelf life is also a major area. We have 40% post-harvest losses. So this is very important area. So farmers are getting more profits. Removal of allergen genes from the food crops and production of pharmaceuticals and antibodies. So these are the among the many benefits which are offered by plant biotechnology. But despite all that, if you see there is low public acceptance. So there are many reasons why public acceptance is low. Still in Europe and Asia, there is less support for GM crops, and there are a lot of debates on GM crops. There are reasons, long-term effects. There are unknown risks and fears, environmental safety issues. Then controversies from NGOs, scientists, and media. So what are the constraints? So the main constraint is transgene escape, that your gene of interest or the marker gene which is giving you resistance against the antibiotic that could escape, and it may progress to non-GMOs. And crop contamination, it could contaminate non-GMOs. There is a labeling issue. So in Canada and US, labeling is not mandatory. So you will not be able to identify that crop is GMO or non-GMO. So there is one example that Starling transgenic corn that is insect resistant. So that is approved for animal feed and humans. So, sorry, it is not recommended for humans because of risk of allergenicity. And this contaminated the non-transgenic corn somehow. And the producing company that has to take all that material back, and there was loss of $5 million in just a single crop. The development of resistance in insect pests, BT crops, particularly removal of the marker gene, that is the main area. Safety and biosafety and ethical issues. People have fear that if we eat GMOs, will it be safe for us? There could be some allergenicity issues. Molecular farming that involves toxic recombinant proteins. So that also is a health hazard to humans and the environment. So other safety issues and ethical issues involved. What will be the effect on population? and the ecosystem that involves animals, birds, insects, unintended harm to the other organisms. Some non-alien species are living around. So what will happen to those species? Herbicide resistance, you may get some sort of super weeds, the action of monogenetic crops with environmental stress. What will happen to the evolutionary effects, evolutionary process in which the crops are following natural selection, and then the food safety. If some transgenes somehow they are transferred to the food, so what will happen if the people will eat that food? The religious and ethical issues in some countries like India, some plants are linked to the religious beliefs, so they don't like that those plants are being transformed or changed. So there is impact on agro-industry production systems. So if we look at different factors, how the rate of gene flow that could be affected in different crops, it is very important to understand that in which particular crop you are working, and what is the genotype of that particularly variety to variety. It could be different. Compatibility is the main issue. Is it sexually compatible to other crops or not? Flowering period, flowering time, overlapping of the flowering time with other varieties. Is it cross-pollinated or self-pollinated variety? So these are all important factors. Similarly, what are the reproductive fitness rate, survival rate, chromosomal recombination rates? So these are also very important. And weather, in which area you are growing that crop, what is the rainfall patterns, light intensity, how the summer and summer temperatures and winter temperatures, all that matters. And particularly the pollen, what are different pollinators, pollinizing agents, I mean insects or other which are transferring that pollen from one plant to the other plant, and about its viability. So these are collectively, these are major factors which will define that how much pollens will be transferred to the and if these pollens are transferred from a transformed transgenic plant, then how much gene transfer will be there. So these are some main pathways involved in unwanted contamination of the gene flow.
Dr. Usman, we can't hear you.
So maybe during seed harvest and transport, when you are transporting seed plants, cross pollination, it could be directional. So these are the main seeds of GMOs, small farmers. So farmers, they have to buy seeds annually. So this is a major issue. And non-labeling of the GMOs, I have discussed before, and market-based dependency and food security, economic losses or exports. So these are also concerns. So there is Cartagena protocol on biosafety that was developed in 2000, that a living organism so any living modified organism that possesses a novel combination of the genetic material, definitely it is obtained through the biotechnology. This could have some adverse effects on the conservation and sustainable use, the biological diversity, and it could be a risk to the human health as well. So all these biotechnology tools involving in vitro nucleic acid techniques, recombinant DNA, or direct injection method, these could be uh, involved. So biosafety is a very wide term which involves biological environment and natural biological factors, biotechnology, pathogens, genetic technology or biological resources, food safety. So I'll focus on the plant-related problems. There are wider applications of biotechnology in agriculture, health, and food. So there is rapid adaptation of the microorganisms. They're getting resistance to the pesticides and antibiotics. So this is a major concern that insects, pests, and microorganisms, they are cause of many diseases, and they are adopting a drug resistance. Rather, it is difficult to control multiple drug resistance pathogens. So serious biohazards and biosafety issues are involving microorganisms. What are biohazards? Biohazards, any species or strain or biotype of the plant or pathogenic agent that is injurious to the plants or plant products. So microbial infectious agents or other biological materials, this could be risk for the human health. So these involve viruses, recombinant DNA, allergens, different genomes. So we must know the properties of the biohazard material, its infection capability, how much pathogenicity or disease severity, how much disease it could create, and is there any cure, vaccine, or therapeutic modality, particularly in animal and human beings, is there available? What is the route of its transmission to the host? Environmental stability. So these are the major areas. So first of all, we have to go for risk assessment, identification of the hazardous properties of the biohazard material we are dealing with, and what activities will be responsible for the exposure to the pathogen, and ultimately, if it happens, so what will be the consequences of the infection. So it is very important that don't underestimate a risk. I can give example of pandemic coronavirus, or overestimate a risk that will involve a lot of cost as well. So need to evaluate factors which are responsible for infection, hazardous agents, and lab procedures. So we need to focus is that because of poor lab procedures or the working attitude of the staff. So for lab biosafety, there are national biosafety guidelines available. Pakistan has also given national biosafety guidelines. So you need to recognize the hazard, assessment of risk, its identification, monitoring, then biocontainment. So we should have proper policies for biosafety and biocontainment, and use of safety equipment, physical containment barriers, and mask, eye protection, lab coats, PPEs. So there are specific biosafety levels which have been properly defined, starting from BSL 1, which is the lower level of the biosafety, and interruption again and again, and BSL 4, which is having higher safety measures for viruses, particularly like corona pandemic. So you have to train your staff for lab safety, usage of microbial techniques. So people should know how to deal with the microbes, and safe use of biosafety cabinets. So how to properly manage the waste being produced in the lab, recombinant DNA technology, because in that we are dealing with viruses and bacterial vectors. Training regarding safety. So if there are some accidental situations, what need to be done, and proper reporting of the concerns, and the team should be working in full cooperation, and there should be self-review as well. So Codex framework has been developed for safety assessment of food derived from GM crops. So you have to document that what is your parent crop, what is its geographical distribution, history, and use of history of the safe use, that what is its computational analysis, what is the donor of the transgene, delivery process, description of the donor, what will be the vector, how you will deliver the transgene, and the characterization of the introduced DNA, and then what will be the gene product, toxicity and allergenicity assessment, then compositional analysis, nutritional analysis, safety analysis. This is also a major concern that it involves many years and a lot of cost is involved before you actually commercialize a transgenic crop. So this is also one of the major problems in transgenics. So these are some general lab safety practices. These involve you need to manipulate infectious fluids if there are spills and aerosols, strict use of scarves, needles, lab coats, gloves. So these should be properly used. Washing of hands, very popular these days because of pandemic. Decontamination of work surfaces, and no eating or smoking should be. These are general principles for lab safety. Coming to ethics and bioethics, ethics that tells you what is good morally and not, what is right and wrong. So when you are involving a biological process, so that is technically called as bioethics and its applications of the biological research. So there are social and moral responses before starting a scientific invention, and that we have to keep a balance, and we must consider what are the pros and cons, what we are going to do. So there should be ethically and socially responsible plant biotechnology experiments, and you must have ethical reasoning, ethical decision making. So what will be the outcome, and how you are going to deal that, how you are going to manage that. Then bioethics in education and science communication. So you have to keep balance in both risks and the benefits. Ethical reasoning and decision making model for agri-biotechnology. So first of all, you need to know what are the relevant facts in that particular place, relevant ethical questions, what could be the ethical questions, who are the stakeholders who are going to make decisions, and what will be the effect of those decisions on the stakeholders, then the underlying values. So what are the biological principles that play a role in the decision? So what will be the possible action and consequences? So what is the best action and why? So all these, generally speaking, all these are relevant questions that you have to ask yourself or to the concerned scientist before starting a proper biotechnology project. So biotechnology has opened a lot of new avenues in agriculture, medical sciences, food and feed safety. So people are shifting from to biofertilizers instead of chemical fertilizers. Renewable sources of energy, biodegradable products are being focused. So still there is reluctance because of raised issues of the bioethics. So this scope of bioethics and biosafety is increasing as the scope of biotechnology is also increasing. So bioethics start from the lab where you are working, and because you are dealing with agriculture, so it starts from lab to the field, to the industry, and ultimately to the government because it involves a wider society. So you must have ethical clearance before starting animal and human experiments. So project review should be properly done with the by the ethics committee for risk assessment, and you have to analyze what could be the consequences and rights and responsibilities who will be responsible for if there is an issue. So similarly, there are environmental ethics considering with the ecosystem and biodiversity. So there are religious ethics. So you have to obey the religious commands, particularly when you're dealing with the animals and humans. Medical ethics, and there are philosophical ethics. So you have to answer all these questions before starting a commercial transgenic project. So now the most important area is transgene containment and alternative plant transformation strategies. So biological containment is relevant, relatively easy if the crop is clistogamous. So there are less chances of gene escape. So then rice and tomato both are clistogamous crops. Total sterility, mostly tuberous and bulbous crops, they don't develop seeds in certain environment. So that involves total sterility. So that could also help in biological containment, engineering, genetic incompatibility. So that could also help in transgene containment. So there are alternative strategies involving plastic transformation. Cisgenesis and intragenesis is getting popular, and more recently the genome editing using CRISPR-Cas system. So scientists, they have calculated on the basis of some evidences that what is the gene flow probability, and they have classified crops based on molecular evidences, pollen movement, and crop biology. So if you look at these crops, including grapes, potato, tomato, so these are the crops which have low risk of transgene flow compared to some other crops. So the alternative strategies involve chloroplast transformation. So as I mentioned that you have to transform the chloroplast. So first of all, you have to develop the expression cassette. So you could use a marker gene or a reporter gene like GFP, and you transform the plastome because of homologous recombination. It is site-directed transformation. So gene will go at proper site that will be decided by yourself. And the transgenic chloroplast, then you have to go for several cycles to develop homoplasmic transplastomic shoots. Other techniques involve transgenesis, in which you are taking gene from somewhere else, promoter from some other organism, and terminator is from some other organism. So in case of cisgenesis, so you are taking all the regulatory elements from the same species or same plant, which are sexually compatible to each other. While in this case, this is actually incompatible gene pool because you are taking different elements from different areas, from different organisms. In case of intragenesis, there could be different genes. Promoter and terminator could be from some other gene, but again, these are sexually compatible. And genome editing, that involves the targeted mutation at specific loci in the genome. So if we compare the genetic modification versus gene editing, so in genetic modification, you are using some gene of interest and you are involving a marker gene. So after several generations, still this foreign gene is in the plant. So this is the major issue that could be resolved in genome editing, in which you are using a gene that will modify your gene of interest, and after that this gene will be removed and there is no foreign gene in the crop. So this is the major advantage. So genome editing using CRISPR-Cas. So you have to develop a cassette, Cas9, and this synthetic guide RNA. So it has the palm sequence, that is 3 base pair. The base side is 3 base pair from the palm sequence. Here you cut it, and there is a seed sequence available in the synthetic guide RNA. So it gives you a double-stranded break. Basically, this is nucleus induced by the Cas9, and that could be repaired by non-homologous hand joining mediated repair or homology directed repair. So that will give you mutants having a gene knockout or style gene mutation or gene insertion. So this way you could develop targeted mutagenesis could be done. So you can compare your crop if it is a non-targeted DNA changes, non-GMOs, and insertion of alternate DNA. You develop a GM crop, so you need to compare that for the altered DNA, what will be the protein and what will be the metabolites formed out of that. So then again, food and feed safety, hazard identification, exposure assessment, and product safety. Similarly, you have to go for environmental safety and risk evaluation, and after that, you would conclude that this crop will be safe or not. So, scope of GMOs is increasing. So, despite biosafety and environmental concerns, there is a large increase in the area under GMOs. This is one of the fastest technologies being adopted by agriculture farmers in more than 70 countries, and 26 countries are cultivating about 192 million hectares. So a lot of billions of dollars are involved in this market, and there are 9 countries in Asia which are involved in the GMO cultivation. So there is a food insecurity in more than 40 countries, 108 million people. So we need to focus on GMOs. The United Nations have developed sustainability goals. So they have commercialized the golden rice, which is designated to fight vitamin A deficiency, and genetically engineered popular, which is good for biofeed production. So how we could ensure food safety? So we need to identify use of organ-specific and pathogen-inducible promoters so that the transgene may not be produced all the time. So it will be produced only when and where it is required. And this is very good area for horticulture crops, particularly fruits and vegetables, that we can graft the non-transgenic sign on the transgenic rootstock. So instead of making a transgenic sign, so we can develop transgenic rootstocks and graft with this transgenic sign, non-transgenic sign, so that the food will be safe. And plastic transformation, that is that gives you maternal inheritance, and there is no chance of gene escape from the full-on gains. So even in plastic transformation, you could express polycystic genes. So one transcript could be translated into several proteins. So that will that has been expressed in tobacco, and 2 PR protein genes have shown resistance against the oomycetes. So genome editing or the alternative technologies could be used, and we need to follow standard biosafety regulation and codex standards. So you can use the nanoparticles, cargo transformation procedures could be used, or Cas9. So you could alter the gene expression, or you could silence the gene, and you could develop transgene-free genetically engineered plants or transgenic plants through the conventional tissue culture. So what are the future biological challenges? Identification of best genes. We need to identify genes which could combat specific insects and diseases. Mycotoxin is a major issue, that its degradation is good for safe development of the safe food products. Hormonal regulation of the responses to the biotic and abiotic stresses. Development of resistance against multiple pests and diseases. And proper risk prediction and assessment. So these are some important web resources which could be used for data collection and updated about the transgenics. Thanks. That's all from my side. If you have any questions, quickly, because I'm being called for the meeting again.
Yeah, thank you so much, Dr. Usman, for your informative lecture. Just a very quick question to you, if you allow me.
Yeah, sure.
How transgenic crops of GM crops can be tackled for ethical and environmental issues? Question from Mr. Aqib Zee.
Sorry, say again.
How transgenic crops or GM crops can be tackled
Yes.
for ethical and environmental issues?
So this is a major debate. I have mentioned in many slides that still people are looking for different methods that how we could remove the marker gene, and nowadays there is a lot of focus on this genome editing technology, so that you could avoid the transgene. So just you modify the gene of interest, its expression, and then you could develop the plants which could be more useful, more safe to the environment. So far, the alternative technologies that I have discussed, this cisgenesis and intragenesis is very useful, particularly for trees, because in trees, there's a lot of issue of development of efficient regeneration involving tissue culture. So this is a major area. So we need, for good transformation, you need a very good regeneration protocol first of all. So as there was a question in the last lecture as well, that this problem of phenolics. So in many woody crops, particularly guava and roses, there are many other crops, mango, so that has a lot of issue of phenolics. So we have to address that, and we need efficient regeneration before starting proper transformation.
Thank you. Thank you so much, Dr. Usman, once again. Thank you so much for your presence, and please allow us that we will share your email ID with our participants.
Yeah, sure, sure. I could take questions by email. You can share my contact number. If somebody is interested, he or she can ask questions on WhatsApp. So for the time being, I submit my apology because I am outstation for a meeting. And anyway, you can share questions, no problem.
Okay, thank you so much. Participants, I'm seeing a few of the conversation between you regarding some of few of you are asking about the lectures of our today's speaker. So let me inform you that the
Sorry
recording.
for interruption. Thank you. I'm leaving now.
Yeah,
Thank you very
sure.
much.
Yeah, thank you. So the recording of this whole workshop will be uploaded on the Comstech YouTube channel, and we will share the link to all of you, so you can download or you can see that recording of workshop at that YouTube channel. We will take a short break of 10 minutes because this was the end of our talks, and we will be gathered again after 10 minutes for the practical session. So please be with us.
Shuru.
So thank you so much. We invite you all again to attend this practical session. I refer to the presentation of Dr. Iftikhar Ahmed, in which he had nicely explained you each and every step of the tissue culture of the horticulture. And now we will show you via these videos each and every step of which is actually required to do for the tissue culture of an ornamental plant. And please do drop your questions in the chat box, and our technical team is here to answer your questions regarding these videos and steps of the tissue culture. So now we are going to start our practical session with this first video.
Tools and apparatus for tissue culture experiment. Apparatus required for tissue culture are forceps, scalpel, and blades, Petri plates with 2 filter paper. All the apparatus should be in spotlessly clean condition. Wrap scalpel, forceps, and blades in paper, brown paper, or newspaper. Wrap battery plates. Put cotton in the jars, which will be used to clean the laminar flow hood. Fill the bottles with deionized water, which will be used to sterilize explant. These things are now ready to autoclave and will be used for tissue culture experiment later on. Media preparation. Preparation of stock solutions for MS media. Weigh desired amount of macronutrients in 1 liter of distilled water, one by one, and store in refrigerator for further use. Weigh desired amount of micronutrients in 1 liter of distilled water, one by one, dissolve them properly, and store in refrigerator for further use. Prepare all the stock solution and store them for further use for the preparation of MS media. Preparation of MS media. Take distilled water from distillation unit. Distillation unit comprises of 2 units, deionized water and distilled water. Take 700 ml of distilled water for preparation of 1 liter of MS media. Turn on magnetic stirrer and put 700 ml of distilled water in a beaker on the stirrer, and also add a magnetic bead into the stirrer to dissolve the nutrients properly. All the stock solutions of MS Media are now ready. First, add 50 ml of micronutrient stock solution into the MS Media. Add 1 ml of micronutrient stock solution with the help of micropipetter. Add 1 ml of vitamin stock solution in 1 ml MS Media. Add 5 ml of EDTA stock solution. Weigh 30 grams of sugar or sucrose for 1 liter of the media. Now add 30 grams of sugar into the media. Adjust pH of the media to 5.7 or 5.8 with 1 molar NaOH or 1 normal HCl. Make up the volume of the media in a measured cylinder. Make up the volume up to 1 liter with distilled water. Weigh 8 grams of agar, which we'll use to solidify the MS media. Add agar in MS media solution. Microwave the media to dissolve the agar completely for 10 minutes. Add phytohormones like auxin and cytokinins according to the plant requirements. Media is now ready to dispense in 250 ml glass bottles. Each jar contains measured quantity of 30 ml of MS media. Close the caps of the jars carefully and tight. And proceed to the autoclave. Turn on the autoclave and open the lid. Place media jars carefully in the autoclave. Close the lid properly and check the outlet valves. Set autoclave program for 30 minutes at a temperature of 121 degrees Celsius and pressure of 15 psi. After autoclave, the media jars will be kept in growth room for 3 to 4 days to check any sign of contamination. In case you find any sign of contamination in your media jar, remove and discard that jar immediately. Contamination-free jars will be used for further experiment. Media jars can be stored at a temperature of 20 to 22 degrees Celsius. Process of removal of the contaminated jars from the growth room. First of all, collect all the contaminated jars and autoclave them. After autoclave, media should be collected in a plastic bag and proceed to discard. The jars are then washed and dried. Cleaning and preparation of laminar flow hood. Wash your hands properly with hand wash or soap before entering into the tissue culture lab. Before starting your experiment, you need to put on your personal protective equipment like lab coat, mask, pair of gloves, laminar flow hood is used to maintain the sterility of the cultures, and in order to maintain the sterility, we need to sterilize our hood first by exposing it with UV light for 15 minutes. Turn on UV light and cover the laminar flow hood as UV light is hazardous for health. Turn off UV light, remove the cover, turn on blower and light. Spray your hands with 70% ethanol before starting your experiment to disinfect them properly. Disinfect the surface of hood with 70% ethanol. Spray the surface of hood with 70% ethanol and wipe all the excess ethanol with autoclaved cotton. Disinfect all the objects with 70% ethanol you are planning to use during your experiment or need to bring into the hood, like media jars, autoclaved battery plates, sterilized tools, put the tools into the sterilizer and turn on the sterilizer. Laminar flow hood is now ready to use for tissue culture experiment. In order to turn off the hood, remove all the waste and used petri plates and media jars. Turn off sterilizer. Disinfect the surface of hood with 70% ethanol. Remove all the excess ethanol with autoclaved cotton. Turn off switch, light and blower and cover the laminar flow hood. Initiation. First step is selection of explant. For initiation step, we need explant. So we are here at the greenhouse to collect our explant. We will select healthy, fresh, disease-free and young leaves as explant. Do not select old, diseased and yellow leaves. Young, healthy, fresh leaves will be excised and dipped into water. And will be taken to the lab for initiation of tissue culture experiment. Explant can be birds, flower, or stem. Cleaning and sterilization of explant. Sterilization requirements are 70% ethanol, 20% bleach, and sterilized distilled water. Once the explant have been selected, take it into the lab and wash under running tap water for a few minutes. Shift plant into the jar or a screw-capped bottle before taking it to the laminar flow hood. Turn off UV light after exposing the hood to the UV light for 15 minutes. Remove the cover, turn on light and blower. Wear pair of gloves and sanitize your gloves with 70% ethanol before starting your experiment. Disinfect everything before placing it into the hood with 70% ethanol like explant, ethanol which will be used for further sterilization of explant, 20% bleach, autoclaved distilled water, discarder. Arrange objects in a way so that there is enough work area for the transferring process and spray your hands with 70% ethanol before starting your experiment. Firstly, explant will be sterilized with ethanol. Shift the explant into ethanol with the help of forceps and shake it for at least 30 seconds. Disinfect the explant by dipping it in 20% commercial bleach, that is sodium hypochlorite, for at least 10 to 15 minutes. and shake it after 1 to 2 minutes. So that all the explant get properly exposed, and at this point, explant will be considered sterilized. After sterilization, explant will be washed with autoclaved distilled water 3 to 4 times in order to remove the traces of ethanol and bleach. After finishing your experiment, disinfect the surface of hood with 70% ethanol. Inoculation of explant into the media. Objective of this stage is to achieve an aseptic culture. An aseptic culture is a culture without contamination of bacteria or fungi. Put on pair of gloves and sterilize them with 70% ethanol. Disinfect the sterilized explant with 70% ethanol before taking it into the hood. Take your tools out of the sterilizer, like forceps and blades. Take out the explant from water and put it on petri dish with 2 filter papers. Cut the edges of leaf with the help of scalpel, as bleach might damage the edges of the leaves. Hold the leaf with forceps and cut it into small pieces with single sharp cut to minimize the release of phenolics with sterilized forceps and scalpel under highly aseptic condition to minimize the contamination rate. Once the leaves have been excised, we will inoculate into fresh MS media. MS media contains different concentration of cytokinin and auxin. Inoculate 2 or 3 pieces into the jar and gently press on the explant with the help of forceps to ensure that they make contact with the media. Cover the jars and wrap them with paraffin. Paraffin will stretch to seal the jar to avoid any kind of contamination. Label the jar properly with the type of plant, type of media, and date. These jars are now ready to shift into the growth room under controlled condition. Disinfect the surface of hood with 70% ethanol after finishing your experiment. Turn off switch, light, and blower. Keep the jars in growth room under controlled condition with photo period of 16 hours light and 8 hours dark at a temperature of 24 degrees plus minus with 40% relative humidity. Shooting, second step of tissue culture after initiation. Objective of this stage is the formation of shoots. Hood was exposed to UV light for 15 minutes. After 15 minutes, turn off the UV light and remove the cover. Disinfect the hands and surface of hood with 70% ethanol before starting the subculturing of pluricomb-like bodies. Pluricomb-like bodies are somatic embroid-like structures formed in vitro during germination from where the shoots and root arises. PLBs can be induced directly from explant, such as shoot tips, flower stalk, buds, root tips, and leaf surface. The indirect regeneration of PLBs can be done by embryogenic callus culture using solid or liquid suspension cultures. Media ingredients are the key factors for successful PLBs regeneration in vitro. Pluricomb-like bodies developed under the leaf surface of explant. As you can see, these are the pluricomb-like bodies developed within 3 months after initiation. Before taking the jars into the hood, disinfect them with 70% ethanol. These jars are now ready to be shifted into the fresh medium for shooting purpose. Before starting experiment, take your tools out of the sterilizer like forceps and scalpel. Arrange your autoclaved battery plates and jar. Always use fresh cultures with no sign of contamination. Use fresh media with additional hormones which are BAP, benzylaminopurin, and NAA, naphthalene acetic acid, which are growth regulator to initiate the shooting. Open the jars carefully and take out the cultures without touching the edges of the jar, and do not use hot forceps as it can damage the culture. These are small pluricomb-like bodies ready to be shifted into the fresh media. Remove phenolics, dried, and dead parts before shifting into the shooting media to avoid contamination. Carefully shift these puricomb-like bodies into the shooting media one by one. And do not use hot forceps as they can damage the puricomb-like bodies or your cultures. Avoid crowding of cultures into the media jar, as these cultures need space and nutrient for their growth. Seal the jars with parafilm to avoid contamination. Label the jars with culture name, date, and type of media. These jars are now ready to shift into the growth room. These jars are kept in growth room with illuminated condition at temperature of 24 degrees plus minus for 90 days. During this time, observe the cultures regularly for any sign of contamination. Keep visiting your growth room after 2 to 3 days to check the contamination status. If you notice any sign of contamination in media jar, discard that jar immediately. This is a contamination-free jar. Keep it for further growth in your growth room. After 3 months, shoots have been developed and they are ready to shift into the rooting media. Rooting is the next step of tissue culture after shooting. The objective of this stage is to prepare plantlets for transplanting by shifting them on rooting media to develop roots. Expose the hood to UV light for 15 minutes. After 15 minutes, turn off UV light and remove the cover. Turn on blower and light. Wear a pair of gloves and disinfect your hands with 70% ethanol before starting your experiment. These are the developed shoots without any sign of contamination. Spray the jars with 70% ethanol before taking them into the hood. These are the well-developed shoots ready to shift into the rooting media. Rooting media contains IAA, that is indole acetic acid, and IBA, that is indole butyric acid. Take out the shoots with the help of forceps and do not use hot forceps as it can damage your plant. Once the shoots have been taken out from the media jar, separate them carefully with the help of forceps and remove dried dead leaves and phenolic parts to avoid any kind of contamination. After removing dried dead parts and phenolics, put them in separate petri plates. Separate carefully to avoid any kind of damage. As you can see, it is a well-developed shoot. Shift it into the rooting media. You can shift a single shoot or a bunch of shoots. Cover the jar with the cap. Wrap the jars with paraffin to avoid any kind of contamination. Label the jars with the type of plant, type of media, and date. These jars are now ready to shift into the growth room under aseptic environment. Clean the surface of hood with 70% ethanol after finishing your experiment. Turn off switch, light and blower. These cultured jars will be kept in growth room under 16 hours of light and 8 hours of dark with 40% of relative humidity at temperature of 24 degrees plus minus. Keep visiting your growth room in order to check any kind of or sign of contamination in your culture jars. It is a contaminated jar and you have to discard it immediately. Keep contamination-free jars in your growth room for further growth. Acclimatization. Acclimatization is the shifting of well-rooted plant into the greenhouse. After 4 to 5 weeks, plantlets are well rooted. The elite and healthy plantlets must be acclimatized in stepwise manner. These plants are ready for acclimatization. Take out the jars from growth room. Before acclimatization, plants need to be washed in running tap water. Remove the cover and take out plant from media jar. With the help of forceps, carefully and wash the roots in tap water to remove the media attached to the roots. Put the plants in petri dish. And separate them carefully to avoid any kind of damage to the roots before taking them to the greenhouse. Do not expose plants to the direct sunlight as plants are highly sensitive. Perlite mixture and peat moss can also be used other than coconut husk according to the plant requirements. Dip the washed and separated plant in fungicide first, then in tap water to avoid the fungal infections because at this stage plants are highly sensitive. Cover the plant roots with coconut husk. Coconut husk is used to maintain the moisture. You can shift your plants in perlite mixture or peat moss according to plant requirements. Repeat the same process for all the plants. Keep the plants in greenhouse and do not expose them to the direct sunlight. Cover them with polythene sheet, which will be removed after 15 to 20 days, as plants are highly sensitive at this stage.
Thank you very much. With this, we come to the end of our one-day online certificate course on plant tissue culture techniques. I would like to thank Dr. Samar for organizing this event. In addition to that, we are also very grateful to our speakers and participants for taking out their valuable time and joining us today. Now, I would request His Excellency Professor Dr. Iqbal Chaudhry, Coordinator General Comstech, and Director ICCBS for his closing remarks.
Assalamu alaikum wa rahmatullahi wa barakatuh. Thank you very much, Hazima and Dr. Sama, for organizing an excellent event and the contribution of very learned resource persons. I can see about 90 people stayed till the end, which actually reflect very high quality and relevance of this certificate course. And I'm sure that this course have contributed in developing understanding of this very important technique. I would like to recommend that the alumni of this course may like to form a group and discuss their mutual experiences. And of course, identify areas in which they would like to have more training. And Comstech would be very happy to organize such training, because I think these training needs to be demand driven, and such training can contribute in development of overall capacity of people in some excellent areas. With this, I would like to thank all of you, all the participants, resource persons, and excellent organizers for this very meaningful and very well organized scientific event. Thank you.
Thank you very much, sir, for your kind words, and for the participants who have been eagerly asking for the presentations and the recording of this lecture. This lecture will be available on Comstech's YouTube channel. I have shared the link for the YouTube channel. In addition to that, all the question and answers posted in the chat box have been answered by the team of organizers. And with this, we come to the end of this session today. And thank you very much for joining us, and hope to see you soon in some other lectures. And there has been some suggestions that people want to have online webinars on stem cells and gene therapy, which Comstech is looking forward to organize soon, inshallah. With this, with this, we come to end of the lecture, and thank you very much for joining us today. Bye-bye.
Thank you so much.
Thank you.